Thank you very much for the opportunity to present these initial results from the Codify study. And I'm a bit trepidatious now of touching any of the equipment at the front here, so bear with me. I'm also aware that I'm the one thing between you and your coffee, so let's get on with it. I've been asked to present the initial results from the Codify study, and this was a collaboration with a large number of investigators across both England, internationally, and within the University of Leeds, the School of Healthcare and the Trials Unit. It was funded by the NHS National Institute for Health Research, and hence I'd like to acknowledge their funding, but remember that the views and opinions are not those of the NHS. And before we get into the core of the presentation, I want to say a big thank you to all the centres and patients without whom this study would not have been possible. And it's lovely to see some of the investigators in the audience. So, as we've heard, diabetic foot ulcers are a significant problem. A large proportion of them can come infected, and because infection is a common feature of the pathway to amputation, then clinicians are vigilant about infection, and when they detect infection and diagnose it clinically, I'm glad William and I are both singing from the same hymn sheet, then antibiotics are usually started immediately with samples taken in order to allow clinicians to then later tailor, should it be necessary, the antibiotics to the up-to-date flora information from the microbiologist. But there's a bit of a question for clinicians. Should we sample these infected wounds using tissue samples or wound swabs? Do you get the same amount of information from those two sampling techniques? Particularly if you think about a tissue sample goes deep into a wound and gets material from in the wound bed, whereas a wound swab collects information from probably a slightly larger area of a wound, but doesn't collect information from deep inside the wound. So, do tissue samples report pathogens more than swab samples by going deep into the wound and collecting the real troublemakers rather than the tourist bacteria sitting on top having a bit of a suntan? Or do tissue samples report pathogens less frequently than swab samples? Or do they report exactly the same amount of information? Or do they simply disagree in that sometimes a tissue sample will pick something up, but it will be missed by a swab? Or on other occasions, a swab will pick it up, but it will be missed by a tissue sample. This was the background to this research being commissioned by the NHS. So the official question that we were setting out to answer with our research was, do wound swabs and tissue samples agree with each other, or do they disagree? So the method we used was a cross-sectional study in which a diabetic foot ulcer, which had been determined by the clinician as being clinically infected, had first of all a wound swab taken and then immediately a tissue sample taken from the same area. And in this way, we could then compare whether you got more information from one technique or the other. In a small subgroup of patients, we collected two sets of samples, and we are doing a small sub-study to compare the regular microbiology analysis, where you send things off for plating and culture, such as these squiggles on the agar plate, and comparing the results that you get if you send it for DNA fingerprint analysis, such as this little picture down at the bottom right. So if anyone's a murder mystery fan, this is the sort of genetic fingerprinting you get whenever the murderer leaves fingerprints at the scene. Because increasingly hospitals have access to PCR machines. Those are machines that do polymerase chain reaction that looks at microbiological RNA. and allows them to determine what bacteria have been in the wound, and rather interestingly, not only the alive bacteria, which have managed to keep breathing from the wound patient to the lab, but also dead bacteria. So in case sampling techniques are good, but actually transport is bad, then PCR techniques might be more useful in the future. So over a period of 19 months across these 25 centres in England, each represented by a red blob. Thank you if you were a red blob. You were one of our recruiting sites that allowed us to recruit our target sample of 400 patients in probably just 2 months over our target time. And our clinical sites assessed formally 680 patients for inclusion into the Codify study. We had 400 patients recruited into the study, and of those, 395 patients provided both information from swabs and tissue samples. So what did our patients look like? And this question's important to set into context any piece of research. In the same way that the HEAL study has got very few inclusion and exclusion criteria, that means that the population included within that study will look like most populations in the UK. They won't have simply cherry-picked the most compliant, the most willing and able patients. We're presenting our patient profile. This allows you to see whether there would be external validity or generalizability of our results. So on the whole, these patients looked like the regular diabetic foot ulcer patient. They were male with type 2 diabetes, and a quarter of them or so had the recurrent ulcer. 50% of the patients had an ulcer that was defined as neuropathic, and almost 50% had an ulcer which was clinically described as being neuroischemic, both neurological and ischemic aspects. A third of them were described as having grade 1 Wagner scale tissue damage, a third grade 2, and the other third deeper diabetic foot ulcer. And you will note that a significant proportion of the patients already had antibiotics on board or had antimicrobial dressings. So our first question was, okay, these patients are all clinically have got an infected wound. Does the swab or the tissue sample find anything? And what we found was that from the swab results in 395 patients, 20% of them, for 1 in 5 patients, the swab results came back with nothing grown at all. Nothing to report, nothing to declare. And this was the same for 1 in 10 of the tissue samples. So whilst the clinician had diagnosed infection, when the swab or the tissue was analysed by the lab, in a significant minority of patients, the report came back as not having any isolates whatsoever. So can I draw your attention to the fact that at least one pathogen was reported from 70% of swabs and from 86% of the tissue samples. And so I'm now going to tell you about that 70% of the swab samples and the 86% of the tissue samples, and tell you a bit more about what was happening in those wounds. So the most prevalent pathogens that we detected are listed here, and they're grouped in different ways reflecting the way in which the laboratory reported to the clinicians in either the diabetic foot clinic or the hospital ward or the community podiatry clinic what was in that wound. There were 79 isolates identified overall, and these are simply those likely pathogens that were prevalent in more than 8% of samples. I didn't want to take up the next 4 slides listing the other 70 or so less prevalent pathogens. So these are the most common likely troublemakers identified in the tissue and swab samples. So, was there agreement between the swab and the tissue for identifying each of these potential pathogens? So I'm now going to take you through and summarise the level of agreement and disagreement and the difference in the levels of detection in swabs and tissue samples. So first of all, the first overview is that swab and tissue reported the same pathogens in only 42% of patients. Swab reports more than tissue in just 8%. So in some cases, taking that swab from a slightly bigger area, pushing down on the wound bed to express the fluid to get it well covered, identified things that weren't detected in the tissue sample. But in a far greater number of cases, in 36.7% of patients, then the tissue sample actually reported additional pathogens that weren't reported in the swab. So this indicates to us that there was only agreement in 42% in that overall summary. But what we've done here is we've lumped all the pathogens together. And so what I'd like to do now is unpack that summary information by looking at those pathogens on a group basis. So looking at some of these bacteria on a group by group basis. So that's what we're going to do next. So for each of the results that I present, you will see the proportion of swab reports that reported that pathogen, and the proportion reported from the tissue samples, and then the difference between these two, and the extent to which we're confident that couldn't have happened by chance by reporting the p-value. So I'll also summarise the agreement and the disagreement. So this is difference at the top of the slide and agreement and disagreement at the bottom. So as an example, MRSA was identified from 27 swabs from the 395 patients who had both tissue and swab samples, and from 31 tissue samples. But it's not the case that those 31 tissue sample reports were the 27 swab results plus 4 extra. Because remember I said before, sometimes a swab picked up things that a tissue sample didn't, and sometimes a tissue sample picked up things that a swab didn't. So we have some excellent statistical brains on the team, and they have worked with us to summarise the results. And if you look at this, it shows that 6.8% of the swab samples reported MRSA, 7.8% of the tissue samples reported MRSA, so the difference in reporting was 1.0%. I chose this example because I could do the maths. And whenever our statistical wizards take that into the dark room and do stuff on the computer, it comes back that the McNamar's p-value, apparently it's a very nice p-value, you'd like him, his p-value comes out as 0.2. So that indicates that there's no statistically significant difference in the reporting of MRSA from swabs or tissue samples. So that's how I'm going to present the results. So that's MRSA. So for MRSA, there was no significant difference between the reporting from swabs and tissue samples. And when you present this as a 2x2 table, for anyone that is unaware of 2x2 tables, it means these 4 cells in the middle, 2 rows and 2 columns. Because I'm Irish, I think I'm allowed to say these tables tend to have about 5x4 rows and columns all over, so I always find it a really strange way of describing a table. This one's actually got, 5 rows and 4 columns. But let me just walk you through this. So this indicates that for MRSA, the tissue sample results, I'm trying to use the mouse so that the video will pick it up, tissue sample results showed that there was no report of MRSA in 92% of cases and a report of MRSA in 7.8% of cases. And when you look at the swab results, they're down here, then MRSA wasn't found in 93.2% of swabs, but it was found in 6.8% of swabs. And when you put these into the 2x2 table, because you've got a fantastic statistical team who'll do that for you, then what you see is that they agreed with each other in 91.9% of cases. That is, the swab didn't find it and the tissue didn't find it, and they both agreed. It's not there. They both agreed that MRSA was there in 6.6% of cases. So the swab found it and the tissue sample found it in these 26. So when you want to summarise the agreement between swabs and tissue samples for MRSA, you add this 91.9% there to the 6.6%. And again, I chose this because I could do the maths. The agreement was 98.5%, and the disagreement was the rest. So you can see the disagreement I've put in bold here. So this poor little one soul here is MRSA was reported in a swab, but not reported in a tissue. And these 5 here is they were reported in the tissue, but not in the swab. So there's disagreement in 6 out of 395. So for each of the likely bad boys, each of the likely pathogens, we summarised not only the difference in the sample reporting, in this case it was 1%, remember, we also summarised the agreement and the disagreement, and whether it was likely to have happened by chance. And when you walk through that process for each of the bacteria, you can get a large summary table, which I'm going to present now. So for the 12 most common likely pathogens, these are the results. So I'll just walk you through these. Remember, these are the groups of bacteria that we found before, either on a genus or a species level. And this is the amount of disagreement. This is the difference in the bacteria reported. So when this is 9.6%, it means that there was 9.6% more reporting with tissue sample than swab. And then this p-value tells you the probability that this has happened simply by chance. So in only 3 out of the 12 likely pathogens here is there no statistical difference. And that's for Staph aureus, excluding MRSA, Pseudomonas and MRSA. So in the vast majority of likely pathogens, there was a significant difference in the report of a likely pathogen, with more being reported by the tissue sample than the swab. So this is the first summary of agreement. We can then add a kappa result, which some of you may be aware of is one other way of looking at agreement between results. And when it's very bright green, it means there's high agreement, and whenever it's orange, there's low agreement. But kappa is useful in some cases. I'm not going to dwell on it today because of pressure of time, but you will see kappa reported as well. But the summary here is the number of people for whom there's a statistically significant difference, and it ranges between 3 and 13.7% difference. So that's one of the key areas of disagreement. The next area that we looked at was whether the number of pathogens reported in each swab or tissue sample was the same. We found that overall, the swab reports came back with an average of 1 likely pathogen, and the tissue sample 1.5 pathogens. And the distribution here is, as you can see, and in some cases, tissue samples came back with up to 6 pathogens reported. So there was a difference in the number of pathogens, with tissue samples reporting more than swabs. And we've summarised here for you the fact that tissue and swab sampling had the same number of pathogens in only 50% of patients. And in most cases, the tissue samples reported additional pathogens over and above the swab samples. We've been able to identify whether the identification of pathogens varied according to the type of ulcer, some clinical characteristics of the patients, and to find so far that this does vary by clinical centre, and that it also varies by the age of the ulcer, with the less chronic ulcers, i.e. the younger ulcers, having less chance of a difference in the swab and tissue sampling. So to summarise, swab samples from clinically infected foot ulcers had nothing to declare in 1 in 5 patients' swabs, compared with 1 in 10 patients whose wound flora was summarised using tissue samples. For the most prevalent pathogens that we identified, isolates were reported in significantly more tissue samples than swabs. And the only 3 areas where there was an exception to this was in the identification and reporting of Methicillin-resistant Staph aureus, methicillin-sensitive Staph aureus, and Pseudomonas. For all other likely pathogens, they were more likely to be reported by tissue than by swab. In just over a third of ulcers, tissue samples reported additional isolates, whereas in 1 in 12 samples, swabs reported additional. So you see, it's not simply the case that tissue always gives you more. In a small number of people, swab also gives you more information as well. So before I finish, I just wanted to reiterate that this study looks at the reporting of swab and tissue samples. And William mentioned earlier that the microbiologist may treat swab and tissue samples differently depending on the information that's provided to them. We're only looking at reporting. Some of the work that we're doing over the summer now is looking at the way in which the microbiology teams actually reported that they handle all of the tissue samples so that we can see if we can explain any of our results simply by the fact that the microbiologists might have used different reporting criteria. We don't know, for example, whether getting more bacteria from a wound swab is because more bacteria were collected from a tissue sample than a swab, or whether it's simply more likely to be reported by the keen microbiologist. And one thing that we haven't yet determined is actually whether this difference that we have observed would make a difference clinically. So we have a few other aspects of the Codify study that we're working on, as well as investigating the extent to which patient and ulcer and dressings, for example, affect these results. We're also looking at level of growth to see if those are reported differently by tissue and swab samples. But we've also got a clinical panel review of the reports from the microbiologist to determine whether clinically seeing the level of detail from the tissue sample would actually make a difference to the subsequent prescription of new antibiotics. We've also been fortunate enough in that we've obtained additional funding to report the prognosis at 12 months, and that will provide information, I'm sure, useful to William's team. And as I noted, we'll compare plating and culture against the genetic fingerprinting to see if this move towards PCR is useful. So thank you very much. I'll just leave the results there in summary, and thank you for your attention. Thank you.